you very much for coming today. We are very excited collectively to be able to share a fantastic story about an area in our lives which is huge. Cancer is a massive generational world issue for us, and it is important because most of us have been touched either directly or indirectly. I'm honoured on behalf of Leslie Chong, the CEO of Imugene, to be able to introduce to you three of the leading lights in immuno-oncology around the world. And it's amazing that we've got them here today, because you wouldn't normally see three people of this level of expertise here together outside of a massive major international conference. So we have Dr. Axel, who's Professor Christoph Zielinski and Professor Ursula Wiedermann. The format today is they will do an individual presentation each, then there'll be a panel session, and I'm sure you're going to have lots of questions. What I would ask you to do is hold those questions back until the panel session, so that then we have a better chance to answer them as a team. But as I said, it's very rare for us to actually have this level of skill and amazing talent in a room outside of a conference. So my job is to step out of the way as quickly as possible, and I've done enough already. So can I please introduce you, first of all, Dr. Axel, who has a list as long as your arm of talents and skills and bodies that he stands on, but there are two that really do stand out. Obviously, as a Senior Vice President at GlaxoSmithKline and the head of their Immuno-Oncology team. Also, co-director of the think tank for Cancer Immunotherapy Consortium. And finally, of course, a non-exec director for Imugene. So, Dr. Axel, could you please come and share your thoughts with the audience?
So good afternoon. It's an honor to be here. Thanks for the kind introduction. The way we've structured this, you'll hear three talks. I will start with the big picture on immuno-oncology. Why is it even worth looking at immuno-oncology as something to invest in? So what's the business case, so to speak? And we hear from Professor Zielinski about the clinical value that immuno-oncology drugs can produce. And we hear from Ursula Wiedermann about the value of vaccines, because ultimately the effort that Imugene is making, it's using the immune system through vaccination to produce benefit for patients. So I'll start with a business case, and who runs the slides? The next slide, please. Okay. So a very simple statement. If you look at that curve here, it's mostly flat for more than 100 years. We have tried to use the immune system to treat cancer for a very long time. We have never had the knowledge and the tools to actually make this a reality and really improve the lives of patients and extend the lives of patients by using our own immune system. Until sometime in the 70s and into the 80s, when we started having monoclonal antibodies, and then we moved to much more sophisticated immune mechanisms, and in the last 5 to 10 years, we really had an explosion. We had several products licensed and put to the market that have generated a lot of value for investors, and that actually produced a lot of value for patients in terms of extending their life. And it's a very recent thing, and there is a lot of new stuff coming now that builds on the last 5 years of immuno-oncology success. If you go to the next slide, what does this look like specifically? As of 2010, 2011, that's the breaking point. Prior to that, we haven't really had any successful medicines in immunotherapy. From that point on, we broke the tides. In the first generation, there were two medicines. Ipilimumab or CTLA-4 was the first that really mattered. And then the tides were changing. In the second generation, you had all these PD-1 blocking antibodies that are now very broadly investigated. There are several products on the market. There's several $1 billion of value per year for the companies that are producing them. They changed really the way we're looking at immunotherapy. And now we're entering the third generation. New mechanisms, much bigger than anything we've seen before, and the way for that third generation has been paved by the PD-1 blocking antibodies in the second generation. The reason I show it to you this way is simply because there's a staged evolution. It started small, it got bigger, and it's going to continue to be bigger. And that is expressed not only in the medications and the benefit that you can provide, but also in the market size that comes from that for investors. So think about it this way. There was no market for cancer immunotherapy up to 2011. Between 2011 and now, we've extended this to about 12 billion of sales per year. And the projection is that by 2025, will be at 60 billion. And this is, you know, there's a certain confidence interval around this, but there's a significant growth curve. This is the greatest growth in the entire pharmaceutical industry of any area. So it's not insignificant. Go to the next slide, please. So I want to say a few words about, in very simplified terms, about the immune system to illustrate why this opportunity is so big. If you look at the immune system in simple terms as adaptive immunity and innate immunity. So what's adaptive immunity? Adaptive means the immune system is picking out the challenge. It sees a target or it sees a disease or a threat. It builds a response against that and then sends out its cells that can fight the disease. There are two types of cells. T cells, which are killer cells, they do the job themselves. And B cells, which are making antibodies. And then the antibodies are producing the response against the disease. For Imugene, we're talking about antibodies. So that's the mechanism Imugene is interested in. And then there's innate immunity, which are also cells, different types of cells that are less specific than the adaptive immunity, but they act faster. So if you now take that as the immune system, then everything underneath are different mechanisms you can use to make a medicine to influence the immune system to fight disease. So cytokines are the signaling molecules in the immune system. Cell therapies is making cells, engineering cells to fight the disease. T-cell vaccines, that's what the field has done for many years. You basically try to stimulate the T-cell side of the immune system to fight cancer. B-cells, this is very new, that's where Imugene is at right now. Checkpoint modulators, that's where the PD-1s and the CTLA-4s of the world fit in. Connecting bispecific antibodies, other types of bispecific antibodies, more small molecules, and you know of that. The pharmaceutical industry has for many years been very successful by making small molecules against a variety of targets. Oncolytic viruses and adjuvants. And adjuvants we've been using to enhance vaccines for a very long time in the infectious disease world. They are very popular and very effective. So if you now look at that big picture here, let's actually go to the next slide. Everything that's green has an approved product in the market. So the 12 billion of market size today for immuno-oncology comes from these areas and predominantly from that one box here, T-cell checkpoint modulators. That's where the PD-1 antibodies are. When you now look at how big the space is, there are a lot of untapped areas. And there's some that are particularly promising. And what we're trying to do here with Imugene is offer you something that is at the forefront of the next wave, of the third generation, using a new mechanism that is very well established. So, and using antibodies against cancer and stimulate them very specifically with focused vaccines. So if you go to the next slide, that color here, that box is where Imugene resides in that picture. Next slide. So a few statements about the business case. So innovation in the entire pharmaceutical industry, innovation or inventing new things is what drives value. If you look at blockbuster medicines or medicines that set at $1 billion a year or more, and approvals of new drugs that have breakthrough designation, which is a designation that the FDA come up with in the US, to say this is a particularly valuable medicine with a big impact for patients. Oncology, here compared to many other areas of the pharmaceutical industry, oncology produces the highest number of breakthrough designations and the highest number of blockbuster drugs. It's a very big difference compared to anything else. So that makes the case, if you invest in an oncology program or in an oncology company, it has the highest probability of producing meaningful returns. You go to the next slide. And here is the growth of that story. The biopharma oncology franchise for the five biggest companies in that industry. In 1991, the oncology market was relatively small because we didn't have all the tools yet. It grew by 2016 to about 27 billion in sales for the lead company, Roche. And this is projected to be 31 billion by 2022. That's just one company, sales for oncology alone. Not talking about the other parts of that pie here. So oncology is the lowest slice here. That alone is 31 billion for the total sales of the leading company. go on. And here is a bit of the growth curve that oncology can produce for pharma. It outperforms basically anything else in any other aspect of these companies. If you look at oncology, the compounded growth rate over the period from 2010 to 2020 for oncology is the blue bar, and the rest of the company is the light blue bar. So oncology is worth more than any other thing these companies are doing collectively. Next please. So a few words now about immuno-oncology. Oncology, to make a bigger picture, everything that targets cancer falls into that bucket. When we now look at immuno-oncology, it's only immunotherapies, which is a subgroup within the larger oncology area. Immuno-oncology, as I said, is young. Only in 2011, also 6 years ago, we saw the first success, and then it started growing from there. So if you look at the big pie here, total growth for oncology is the light blue curve, and on top is layered now the added immuno-oncology value. It only starts counting here as of 2011, 2012. The compounded growth rate for oncology at large, the non-immunotherapies, is about 12%. It's about 43% for immuno-oncology, substantially more. And you can see that not only in financial terms, you also see it in terms of benefit. The survival improvements that immuno-oncology agents can produce are equally bigger compared to non-oncology agents. Go to the next slide. Obviously, an industry that grows so fast also needs to get access to innovation, not only through in-house discovery, so making your own medicine. It takes a 10-year or more time span to make your own medicine. If you want to get into that space faster, especially the big pharma companies like to do that, you buy medicines or buy technologies that small biotech companies are putting together. So in order to do that, you have to do business development deals. So here's a comparison of the immuno-oncology deals versus the non-immuno-oncology deals in oncology. So overall, there are more deals done in non-immuno-oncology than there are in immuno-oncology. But in terms of valuation, the smaller number of deals in IO is worth more than the larger number of deals in non-IO. And that's just the beginning of a trend that I believe will continue, and you will see there will be more and more value placed on immuno-oncology because we're just generating more value for patients this way. There is more value in immuno-oncology in general. Next slide. And here is a bit of a distribution, how early or how late in the development cycle of a given drug are the immuno-oncology versus the non-immuno-oncology activities. The simple message here, if you break it down from marketed products to approved products to clinical programs and preclinical programs. For immuno-oncology, the majority, which is shown here in yellow, is about 63%, which is preclinical for immuno-oncology. The reason for that is very simple. It's an early area. Right? We said in 2011 it began, we only had 6 years' time to really develop all these new immunotherapies. So many of them are not in the clinic yet. And all the deals that are being done are often done on non-clinical programs. That's remarkable because the pharmaceutical industry usually likes to do deals on clinical programs. It's much clearer if something is working if it's in the clinic than if it's not. And on the flip side, and that makes the same point, for non-immuno-oncology programs, the vast majority of deals, more than 60%, is actually done on clinical programs or approved drugs on the market. So that tells you immuno-oncology, even though it's early, attracts a lot of attention and generates a lot of value early. Next slide. And this is a snapshot. I'm actually not going to go through this in depth. It's only supposed to make one point. If you look at the time axis from 2015, this is the entire year 2015, a lot of immuno-oncology deals have been done. These are big pharma companies partnering or buying the technologies of small biotech companies like Imugene, right? Producing something new, pharma needs it, so pharma buys it. And you see some valuations here, 130 million, goes up to 500 million and way beyond that if you have a compounded valuation for any given deal. So there's an upfront payment usually and then there are milestones following that. These are very high valuations, especially if you think about many of these programs being not in the clinic yet. Go to the next slide. And here's the same picture for the year 2016. I don't have it for 2017 yet. It takes a little bit of time to put this together. The next slide. And here we have how these research programs break apart in the current industry. If I break apart what kind of immuno-oncology therapies are being investigated preclinically or clinically, and yet I've shown you the chart at the beginning, what's the landscape, what does the total landscape of the immune system look like? The vast majority here are cancer vaccines. Let me see where that is. Here. Cancer vaccines are this big size of the pie of the investigated types of immunotherapies. So what Imugene is doing is producing a cancer vaccine, but there's one important differentiation. Almost all of these cancer vaccines are T-cell vaccines. They're using your T-cells, the killer cells, to fight cancer. Imugene is using B-cells. There's a very unique aspect about the B-cells. We know they work very well for infectious diseases. Basically, any vaccination that you give your kids to prevent disease, use B-cells to make antibodies. We just haven't, because the field is so young, we only had 6 years' time. We haven't come around to use that yet for oncology, for treating cancer. So I think there's a lot of confidence in vaccines, as you can see here. The B-cell story is just beginning. There's an opportunity for us from a scientific perspective to make a contribution. There's an opportunity for you, if you so choose, to actually invest in that and create an opportunity. Next slide. So I'm coming back to that picture I've shown you in the beginning. This is where Imugene is with its unique B-cell vaccine story, and it fits very nicely in the overall immuno-oncology landscape. And it is well differentiated. It's very different than what most other companies are doing. And I think we're lucky that we have access to this technology. If you go to the last slide, please. And this is the summary, basically, of what I've just told you. There's a lot of innovation in oncology and in immuno-oncology, a lot of new technologies, and this is the fastest growing area in the pharmaceutical industry. Faster than any other by a long margin. It's heavily sought after, and the deals that are made there are very highly valued. The focus previously was mostly on T-cell based therapies. We're now beginning to see there's interest in non-T-cell based therapies, particularly in vaccines, and that creates, for the B-cell story that Imugene has, it creates a unique window of opportunity. And I'll let you judge how much of an opportunity that should be. And I think you need to hear a little bit more about the clinical value that can be generated, and actually what do vaccines do with the immune system. So you can put a little bit more meat on the bone for this story. So that's, in simple terms, that's my presentation of the business case. And then I'll give the word to Christoph Zielinski to tell us a bit more about the clinical observations that we make with immunotherapy in patients. Thank you.
Ladies and gentlemen, good afternoon. Thank you. I'm very happy to be with you here, coming from the other side of the world. And it feels like the other side of the world because I have 10 hours time difference, so I try to stay straight. Should something happen, it's not alcohol. I'm a medical oncologist, and being a medical oncologist, I have started a couple of years ago. It was not very many, at 41. And we were starting out with chemotherapies. At the time, we only had chemotherapies, and that was the only thing that we had. In the majority of cases, we only had one chemotherapy or one chemotherapy combination per disease. And the whole field has been revolutionized during the last, I would say, 5 to 6 years. And that is due to immunotherapies and immune checkpoint inhibitors. So a complete change in treatment paradigm that we have with patients with tumors. Now this has led to an enormous difference in how we're treating patients in the majority of cases. by mobilizing the body's possibilities to kill tumor cells which they are surrounding. Now, if I can have the next slide, please. I would want to show you a video now, which the group that I'm sharing, it's a Central European Cognitive Oncology group, has produced, and it shows you how this whole thing works. We have to maybe turn out the...
Yeah, the first few seconds are quiet, so...
Right, so I'm quiet, so everybody's quiet.
The cancer immune cycle involves the local tumour microenvironment, the regional lymph nodes, and the communication between those sites via the lymph and blood vessels. Only if all steps of the cancer immune cycle are successful, an effective immune response can be generated against the cancer cells. However, the cancer immune cycle is tightly regulated, and several cellular interactions in the tumour microenvironment influence the intensity of the T-cell-mediated anti-tumour response. The local tumour microenvironment comprises several important immune cell types. Dendritic cells take up tumour antigen and transport it to the regional lymph node for T-cell priming. T-cells are the main mediators of the adaptive immune system and kill tumour cells via release of cytotoxins. Macrophages are the main effector cells of the innate immune system and have various functions such as phagocytosis and antigen presentation. Tumour-associated macrophages are present in the tumour microenvironment and can express the immune suppressive factor PD-L1. PD-L1 expressed on tumour-associated macrophages can bind to the inhibitory T-cell co-receptor PD-1 on T-cells and thereby inhibit the T-cell function. Activated T-cells initiate tumour cell death by emission of cytotoxins. However, expression of the immune suppressive factor PD-L1 can prevent this. PD-L1 expressed on the tumour cell can bind to the inhibitory T-cell co-receptor PD-1 and thereby inhibit the T-cell-mediated immune response. PD-L1 expression on tumour cells and tumour-associated macrophages results in an immune evasion of the tumour cells. As a result, the tumour can grow unharmed by an immune response. Therapeutic monoclonal antibodies that bind to PD-1 or PD-L1, so-called immune checkpoint inhibitors, prevent the interaction of these immune-suppressive molecules and thus unleash an effective T-cell-mediated anti-tumour immune response. Only if all steps of the cancer immune cycle function properly, immune checkpoint inhibitors targeting the PD-1 axis can unfold their anti-tumour function in the local tumour microenvironment.
Right, so you see how this works. It is a major step forward, and one of the most important things that has evolved during the last couple of years. However, next slide please, we are only at the beginning. This is only the end of the beginning and definitely not the beginning of an end. Why is this the case? It's the case because the area is expanding so enormously. It's expanding enormously and quickly. Dr. Huss showed you this before. And when you look at the whole immunoregulatory cycle, meaning that the immune system is able to eliminate tumors, we do have a series of steps which we really use every day. And that starts with the release of cancer cell antigens, because the immune system works like face-to-face recognition, right? So if I'm standing here and you heard before who I am, you would recognize me, right? So that's how the immune system works. So it's very important that the immune system recognizes these tumor cells as such. And then this turns on to cancer antigen presentation, because it has to be sort of presented to the immune system. Further, it goes on to priming and activation. That's what we saw before. Trafficking of T cells to tumors, which is important because the tumors have to be infiltrated by these effector cells. Otherwise, it doesn't work. This is like a parking place without cars. And then infiltration of T cells into tumors recognition of cancer cells by T cells, and then only the killing of tumor cells. So these are very intricate mechanisms, and I just want to draw your attention to the fact that right now we only have this here to influence and this here to influence. So we have CTLA-4 and PD-1 and PD-L1, where all the other things are still coming and are in the cooking, and in fact are in phase 1 or phase 2 trials. And Imugene is trying very, very much so to include not only a lot of different kinds of approaches like vaccines, where we try to really redefine the recognition of the antigen of it, which certain tumors have, and redefine how this is truly happening, that these antigens are being recognized by the immune system, but also we are expanding right now, and we're on the verge of expanding our understanding how to ameliorate this to make, so to say, a super vaccine by the addition of certain immune checkpoint inhibitors. So this whole thing is truly evolving extremely well, and I'm very grateful to be able to be part of this. The next slide, please. So you see the CTLA-4. The next slide shows you, right, we'll show you this here, and that is the story. Now, what happens in this entire situation is that we are having a certain amount of response. We have a certain amount of response with immune checkpoint inhibitors and immune checkpoint blockade, which then starts very, very well, but at the end, it ends with about 20% response on long term. Nevertheless, we do have enormously impressive responses with 2/3 of patients with metastatic malignant melanoma, which led to imminent death within 6 to 9 months a couple of years ago. Now 2/3 of patients are surviving at 3 years, which is tremendous. We have similar results with non-small cell lung cancers, where we have certain subpopulations of patients surviving up to 50%, up to 5 years. So this is really something which happened only during the last couple of literally months. And we do have a longer tradition in this very context, where we do have the HER2 protein, which is present on different parts of the body, breast cells, breast cancer cells, gastric cancer cells, etc., which we are particularly interested in. And also this targeting led to the fact that women who had HER2 positive breast cancers died within 6 to 9 months a couple of years ago. And now it's no surprise to see these women survive 48 plus months, which is quite remarkable, and that is the molecule we're most interested in. So what we are trying to achieve is that combination of different kinds of immune intervention is then better than only the simple, or quote unquote, only the simple kind of immune intervention. The next slide, please. So what you can do is, of course, you can do a lot of things. You have this active immunotherapy, and you can combine this. You can combine this in a way to ameliorate the response and to make things better. You can combine this with other immunotherapy, and you will hear from Ursula Wiedermann how vaccines can play a role here, and particularly in combination with immune checkpoint inhibitors. You can combine it with chemotherapy, which is extremely exciting, because we know the chemotherapy is in fact working very much like immunotherapy, and only after very many years we started to understand that with chemotherapy we're doing a major part of immunotherapy. You can combine this with radiotherapy, because again, radiotherapy releases these antigens and thereby makes the tumor recognizable by the immune system, and in the end with the various kinds of targeted therapies, which are a little bit more tricky, but again, quite a good thing to do. So this is exactly where Imugene is moving to. We're starting out with a molecule which is right now in clinical trial. We're starting out to generate further and other steps in order to make, so to say, if you would wish so, super vaccines, which would then combine all these aspects. The next slide, please. comes to the fact that particularly other immunotherapy is something where we want to draw some conclusions and come to the point that with the combination with different kinds of immunotherapies, particularly with cancer vaccines and the not prophylactic vaccines, but rather the curative vaccines, we could ameliorate treatment outcomes in such a way that in the end we would be able to induce long-lasting responses towards emerging tumors. So thank you very, very much, and it is my pleasure now to hand the microphone over to Ursula Wiedermann, who will explain where we might be going to. Thank you very much.
Yes, thank you very much. I have a similar situation as Christoph, also coming from far away, and I'm still quite awake. So, so what I would like to tell you is really the concept we are now bringing in the clinic, namely a vaccine against certain tumors, and in particular those expressing HER2, as Christoph Zielinski just explained. So, next slide, please. You heard a lot about immunotherapy and all the different possibilities to interact with the immune system. And this is actually what I would like to talk about today, is what types of vaccines are available and how they can be active. In particular, the most important is that the vaccines work because we activate the patient's own immune system to produce their defense mechanisms himself or herself, and not being dependent on giving already produced antibodies or produced substances to do the job. Next slide, please. So the question is, why do we go for active immunization? Why do we go for vaccination in cancer patients? There are two concepts. So everything that was talked about until now is passive immunotherapy, meaning that the patient is getting already preformed antibodies that will do the job in the body of the patient. The other possibility is, like it was already explained, is when we talk about vaccines, then actually you give parts to the patient, and the patient have to work himself and building up the immune response that will then be active. And the question is, what is better, or what is the advantage of doing your own thing? Next, please. So that mainly lies in two situations, namely when you get antibodies already that are produced and give this to the patient, then you have very quickly a good response, but these effects you're achieving here, they will soon decline, because these antibodies, they will be degraded and then it's gone. So in other words, for the patient, this means you constantly have to give again and again and again these antibodies to have the effects. When you have to do your own business, you inject here the vaccine, then the body is building up these antibody responses by himself, meaning there is in the beginning a slow phase, but then you come to a point where the patient himself continuously produces this immune response, and this immune response will last for a long time. And importantly, what is also very particular for our immune system, it has a memory. So that means it will remember, as Christoph also said, knows already this trained and knows what it shall be produced, and this can last for years and years. So that is quite, from the kinetic point of view and for the training for the immune system, very good to be more active against the cancer himself or itself. Next one. So when talking about the vaccine that is now being distributed by Imugene and being tested in the clinic, is particular a vaccine that is directed against HER2/neu. That is, as it shows here, a receptor on the surface of certain cells, like breast cancer or gastric cancer. They can overexpress this receptor that is responsible for, if it is overexpressed, then cancer cells will proliferate and divide, and the cancer will develop very fast. So in order to get this a treatment against this cancer, you all probably have known already that there are antibodies available. These that they will, like Herceptin, you inject the patient with these antibodies, and then you, these tumor cells will be defended. Next one. Next slide, please. And they work like this, that these antibodies, they will interact with this receptor so that signals by the receptor cannot be transferred to the inner of the cell, and thereby the proliferation and growth of tumor cell is being inhibited. So that's principle, the concept, and what we were thinking is that such a concept would be great if actually the patient is producing these antibodies himself and does the job by itself without being dependent on giving constantly these antibodies to the patients. And that is what we are doing. Next, please. Next slide. So the advantage, if you have these antibodies, then you have a good effect. You can actually interact with the growth. You will stop the tumor growth. You will even be able to activate somehow the immune system, be like a flag for the immune system so that other cells come there and defend. But as I said in the beginning, if the patient doesn't produce the antibody himself, you constantly have to give them again. Next slide. And this is what we want now to do, that the patient is doing himself, and we have created this vaccine where we actually have identified these parts of the receptor where antibodies can be directed against. So we use these parts of the receptor for immunization, as it is, we call it a vaccine antigen, in order to be quite immunogenic, so that the immune response of the patient can react very well. These epitopes have been fused together to a huge peptide. Then it is bound to a carrier, so this is also formed to be more easily recognized by the immune system. And then you add also an adjuvant that gives another hint to the immune system, and that is the whole the formulation as the vaccine looks like. Next one. And if you do that, what happens? Please, next. So you inject this to the patient. Then, as you heard, these B cells are the ones that are the producers of these antibodies. They will be activated, and these antibodies will be produced. They will be released in the circulation, and they will find that this surface of the tumor cells will bind to this. Next one. And then at the end, the result of this is, as we see in passive immunotherapy, the tumor is defended and shrinks. But most importantly is that this will be a situation that will remain, because we train the own immune system to do this, and you will have involved all organs, lymph organs in your body that are responsible for producing these antibody responses, and you will finally produce so-called long-lived plasma cells, so that this effect and this machinery will be kept on for many months and years. Next one. So the next then is what Christoph Zielinski said, is that we have this specific tumor-specific therapeutic vaccine, and then you have on the other hand these checkpoints, which are some sort of regulation in our immune system. So actually for natural cases, these checkpoints are there to control whatever immune response is going on. But in the tumor defense, this is not so good, because you actually want to have the T cells to be all the time active, and this is why you block this interaction here with the T cells to have them constantly running, more or less. And what the idea is now in our system is, next please, is actually, if you have a therapeutic vaccine like our vaccine, you can even more enhance, or whatever vaccine can be enhanced, if you also activate at the same time the T cells. So you combine actually these checkpoint inhibitors with a certain vaccine to get enormously strong immune responses. And that is the next goal we are going for, and there is indication from the clinic already. Next, please. Where this actually has been done, you all heard about human papillomavirus vaccines, which is currently a prophylactic vaccine. But there is the chance also to use this vaccine already in patients that have already the disease. And it has been shown if you combine this with a checkpoint inhibitor that you really get a very active and even better vaccine going on. And that is a very important concept for the future of therapeutic vaccines, how to make them even better and broader and long-lasting. Next one. We have tested this already only in mice yet, but we have tested this principle with our vaccine, with our breast cancer vaccine, and antibodies against the checkpoint. And what we see is that indeed we can have these very strong immune responses going on, and that is obviously a very good combination to go for the future of next vaccines. Next. And what importantly for the future, how to develop so that we not only have this one vaccine that is going into the clinic now, but have new candidate vaccines. We have developed a sort of platform how it is possible if you have antibodies that are tested in the clinic and are being given to patients, how we can actually identify the binding sites on certain proteins, where will they bind, and identify these, screen for these areas here, and then we can produce ourselves, next one, these molecules or these peptides actually, and can use them as cancer vaccine antigens. They will be, like peptide vaccines. They can be combined with carriers so that they are very immunogenic, and that's how we can constantly produce new vaccine candidates, and that's the platform we have established. Next one. So that just shows you with any known or of interest antibodies that can have biological activity, we have now the possibility to use this for identification of the right antigen to identify peptides and create peptide vaccines, so that we have continuous flow of a platform production for vaccines, whatever is of interest. So that what we have established at the other part of the world, and I would like to thank my co-workers next one. They are doing actually the job in this area and keep us all busy with new ideas and new products. Thank you very much.
So thank you very much indeed. I think we're now ready for the panel session. So if our esteemed panel want to come up here, and then obviously I'm hoping that you've all been scribbling down lots of questions. I mean, for me, I think it's fascinating. We've had that journey from why oncology, and specifically immuno-oncology, is an area which has attracted huge amounts of investment. We've also understood how it works in principle, and we started that journey as to specifically what Imugene are doing around the HER-Vaxx vaccine and how that could work. So, can I open up the floor to this esteemed panel for your questions?
No questions. I think calls from you all.
What are the chances of producing a broad protection medicines, at least out there?
Well, actually, I think the vaccine we have been showing is not likely that it will cause a lot of autoimmunity. Of course, you have to redirect the immune system to go in the direction of immune response that is more pro-inflammatory, to have better defense mechanisms. But the thing is, and this is why we want to go for this vaccine, you don't want to have such a high level of immune.
When you answer, whoever's answering, we can record it onto the video afterwards.
Okay,
sorry about that.
So when you have an active vaccine, you will not have such a strong immune response from the beginning, but it really builds up after the time, and you probably have a more controlled immune response that is more particular interacting with the tumor itself and not having so many side effects as we know from passive immunotherapy with certain checkpoints where this is really an issue. So I see that.
That's something here. It is very interesting. This question is very interesting and is clinically extremely relevant, of course. Now, what we know about HER2 and where the vaccine is directed at is that this is a protein which plays a major role during embryogenesis. So it's interesting to realize that you can give chemotherapy during pregnancy to the pregnant woman, particularly the second and third trimester. But you're not allowed to give trastuzumab, which is the antibody against HER2, in this situation because this really hurts the growing child. This stops after differentiation, after delivery. HER2 is not a protein that is around. So we can exclude this induction of autoimmunity. With immune checkpoint inhibitors, this is very interesting. We do have the impression that there are two different kinds of immune checkpoint inhibitors. One is anti-CTLA-4 and the other is anti-PD-1. And as anti-CTLA-4 plays a very crucial role in the recognition between the antigen presenting cell and the T cell via the antigen interaction with the T cell receptor, and this is a very crucial and very central role. By giving an anti-CTLA-4 antibody there, you really do generate autoimmunity, and you know in the clinic that this is a major problem. So you, depending on the time when the drug is being administered, you can really predict after 3 to 4 weeks you have the cutaneous side effects, after 6 weeks you have colitis and diarrhea, after 8 weeks you have hepatitis and hypervisitis, and all this is really autoimmune. Now, with anti-PD-1 antibodies, which are really pushing the CTLA-4 antibodies really aside, you have a rather very classical kind of interaction between the tumor and the T cell. So this is not a crucial molecule. PD-1, PD-L1 is not a crucial molecule, and therefore you have only very limited amount of autoimmunity, if at all. So these are drugs which are tolerated very, very well. The maximum that we see is could be cutaneous side effects. There is a certain amount of pneumonitis, which occurs in about 2% of cases, but not more. And therefore, because this is such a specific kind of interaction, we can exclude this drug. Sorry for the long answer for a short question. I love these short questions. These are wonderful because they allow you to go on and on.
Another question from the room. Gentleman just on the left here. Go ahead on the microphone. I'll repeat the question.
So, when you're using your vaccine to try and stimulate an immune response to HER2, is HER2 considered a self-antigen? If it's
Sorry, if you repeat the question,
absolutely.
For the tape.
So, with your vaccine, you're looking to stimulate immune response against HER2. You've mentioned that HER2 is mainly expressed in embryonic development. Is it expressed in the adults, and is it considered a self-antigen that you're trying to stimulate an immune response to?
It's not. It's only on differentiated tumors. So it is.
Thank you. Only because you were referring to this, what I said, if I take the liberty to answer this. This is not. It does not occur. It is not re-expressed in adults or during normal adulthood. It is expressed on certain amounts of tumor cells and in certain cells only. So, in fact, HER2 was understood that this was one which made breast cancer fall apart as an entity. Because we were always thinking that tumors are an entity depending on the anatomic location. In fact, this was our first understanding that breast cancer is not breast cancer as such only because it occurs in one tumor, in one tissue. So breast cancer has fallen apart in about 8 or 9 entities, one being the HER2 new overexpressing. The others do not overexpress this. So this is a very specific finding in very specific tumors, and this is not a ubiquitous antigen at all.
It is a self-antigen. But it's to answer the second part, it is a self-antigen. So if you are going to produce a vaccine, you have to have a formulation that is able to activate the immune system to overcome self-tolerance. And that is actually what we have been doing by creating these peptides, only not using the whole extracellular domain of the protein, but only peptides, combining them with a carrier system, combining them with an adjuvant. These are all important parts of the formulation to make the vaccine able to produce an immune response against a self-antigen.
So I just added another nugget to this. HER2 is what we're beginning with, right? The clinical program for HER-Vaxx is focusing on HER2 and anti-tumor vaccination against HER2 carrying tumors. Now, what you've heard is there's a whole pipeline of other projects that use the same technology to make vaccines against new targets. They can be like PD-1, as we've heard, which is a big immunotherapy target. And there can be many others. So HER2 is our test case and the first product in the clinic and maybe the first medicine, but it's not the only one. There's a variety of others to follow, which is what makes this so valuable. It's not just one project, it's multiple projects using the same technology.
Just on the business model, there was an Economist article on the 14th of September relating to developments in cancer, and also mentioned in respect of the one-hit vaccines which have got memory, which was demonstrated here, that some of these injections, as costed in that article, were $475,000 US. I'm just wondering how the business model develops when you have a one-hit memory response rate, and that's all you've got. I can understand how mass vaccination for polio are logical, but we've got individual cancers and all sorts of things. The second question related to that is, how do you make money and how do you make your return when the combination therapies are who's responsible for the success rate, the long-term survivability, if you've got multiple graphs of 20%, let's say, longevity stacked on top of each other, and who gets the credit and the money for that? So just on those two questions, how can it all be afforded for a successful economic return for the company? And secondly, when there's combination therapies like you're recommended, who takes credit and the dollars from that?
Yeah, so these are interesting questions, and somehow the economic model for pricing cancer drugs at large or then immunotherapy drugs has to change over time. We are overpricing drugs right now, and I'm the first to say that, and I work in the pharmaceutical industry. This will over time shift. We cannot continue to add on cost without adding on benefit. In some places, we actually have already arrived in an area where we're pricing for value. So that means if you deliver a large amount of value to a patient, it's also acceptable to price it high. But it isn't universally true. So clearly, what we're aiming for is be transformational, so offer big benefit, and sustainability, so long-lasting benefit. If we achieve that, then this takes care of itself. The pricing model so far has always been quite favorable for any effective drug. And what we're looking for is by having targets like HER2 that we know work in cancer, or targets like PD-1 that we know work in immunotherapy, that we price this accordingly. But you know, the company is not yet in a place where we can say we can put a price on a medicine. We don't have a medicine yet. We're still doing research to get to that point. So we will get there depending on where the market is at the time when this happens, right? So
I wanted to add one thing to it because I'm sitting there on a daily business, and we're trying very much to reduce or to control for prices of drugs. Frankly, very, very many drugs are not priced according to the value, but are priced according to what the society is ready to tolerate. Just want to bring to your attention that CAR T-cell treatment, which is directed against leukemia and lymphoma treatment, right now has been registered and will have the price of $450,000. There's another drug which is called Ibrance, which is produced by Pfizer, is a drug which is being given in addition to anti-endocrine treatment in patients with metastatic breast cancer. This drug costs you for 1 month tablets 8,500 euros. So this is about 10,000 Australian dollars, right? And I find, personally, companies which are overpricing these drugs in such a way, much more at a hazard than those who are tending to generate a price which is tolerable. Why do I see it that way? It's because society will not be ready to pay such a high price on a long-term basis. And it's like Dr. Ho said before, where with immune checkpoint inhibitors, we're already there. Similarly to what happened with the HIV drugs, there's a good example. Another example is the hepatitis C drug, which was overpriced like crazy, and then sort of had to be reduced in price. We have it with immune checkpoint inhibitors. We already have right now regulations in place, at least in my country, where we have a risk-sharing model, or if you respond to treatment within 3 months, the hospital takes the costs. If you do not respond within 3 months, the company takes the costs. If you respond up to 6 months, then it's 50-50, and only after you respond after 6 months, the hospital takes the costs over. Because this is such an expensive drug that you can imagine, for instance, in malignant melanoma, for the first year, which is quite particularly in Australia, frequent disease, by the way, the cost for metastatic malignant melanoma, $200,000 in the first year. So I find companies extremely at a hazard which are producing such high price products. This will not be sustainable on a long-term basis.
So, of course, your question somewhat went towards, is this going to be profitable, what we're doing here? That's what I understood. And, of course, looking at what the numbers I showed you at the beginning, oncology has always been profitable. Whatever pricing environment you found yourself in, and this has shifted over the last 2 decades or so quite substantially. I expect it will shift again, but the probability that we will not be profitable with a meaningful medicine is very, very small. So I don't think that will be a problem. And when you get to combination therapy, we have the opportunity here, by the way the vaccines are made, to put 2 medicines into 1 vaccine, or maybe even 3 medicines in 1 vaccine. So you don't have to price them individually and contribute to this enormous cost that comes from putting expensive things together and make them even more expensive, but rather create 1 vaccine that targets 3 things at the same time and price it like 1 medicine. That also contributes to making the pricing situation better. And such a medicine would be highly profitable because it offers a lot at a reasonable price.
Next question. One at the back. Sorry, let me just I'll probably direct it to Leslie first, because you haven't spoken. But I'm sure the rest of the panel will talk. Going to Axel, what you just said earlier, that obviously we're using the same research and the same science to have other targeted drugs. As investors, when can we expect something along those lines?
Well, so Imugene is an incredible company, and we want to announce when the first cohort is complete, and so that you have a full picture as opposed to piecemeal. So when we release to the market is when you'll have that data.
I'm talking about the next one, though.
I'm sorry, the next what?
The next peptide combination, the next.
Right. So we had an incredible strategy meeting today, and that news is soon to follow.
Okay. There was a question just here.
Thank you. Mark Bradley, Morgan Stanley. I just wanted to thank the panel. I mean, it's a real privilege that Imugene has all that scientific power up there. But I also wanted to just beg forgiveness to ask the financial markets sort of a question. And perhaps to Axel, obviously done a lot of interesting research into comparing the different valuations and deals. I was just wondering how you considered Imugene is being judged in a valuation sense. It's difficult for us to sort of assess where the value of your research is, compared to what other people are doing and its potential, etc.
Okay, so I'll take that and take this as my opinion, because that's really all anyone can offer in the financial markets. I think Imugene, given that we're in Australia, we're not in the United States, and valuations of biotech companies is very different in the United States. You can raise a lot more resources, and you can produce a lot more value faster in the US. All the deals I've shown you are US-based deals. Then you can do that here in Australia. I think Imugene is, in a global biotech community, highly undervalued, despite the fact that our stock price has actually tripled over the last 6 to 12 months. So we have generated a lot of value, but it's still much smaller than it could be. So I see upside. There could be a lot more. The science, as it evolves, as we produce more data, should actually lead us to continued growth. I hope that answers the question.
Professor Ursula, this is a question for you. Just on the vaccines mechanism of action, I did see the diagram where you showed that Perjeta and Herceptin both bind on different sites onto the tumor, but when you're giving your B-cell vaccine, that actually produced binding sites throughout that stream. So do you think there's a significance of that to get better responses? Can you enlighten us on that, please?
Thank you very much for this question. Yes, actually we do, because what we are hitting with this vaccine is that we have the possibility of interacting with the different binding sites of 2 different antibodies. So what we expect is that that this vaccine can generate antibodies that are more or less a summary of the effects you get by single monotherapies that are going on. So that's, I think, these polyvalent vaccines we are having, I think, is an important add-on to existing therapies now, plus the fact that you can continuously produce these antibodies. So I do believe that it's a very good selection of antigens, and I think it's the way to go, that you don't stick to only one epitope, but that you include several epitopes to have a broader effect.
So we've got time for one more question, so I'm hoping this is going to be the barnstormer. One more question from the audience. Go. The pressure's on you, my good man.
This is directed toward Axel. Thank you very much to all of you for all your presentations today. Is in relation to the number of trials that are being done on vaccines and the number of T-cell vaccine trials and the number of B-cell vaccine trials, just in order to get a understanding of competition in the B-cell area.
So you can actually...
No, that wasn't me. So did you finish your question? Yes.
So, in general, for immuno-oncology trials at large, and I'll come to the vaccine piece in a second, there's a lot of competition. There are a lot of trials. There's a lot of new technologies that are being tested. Some are really good, some are not so good. That's usually true in an environment like this. So, yes, there's competition. Then when you look at the vaccine space, that's about 40% or so of the total immuno-oncology research endeavor. The vaccine trials are all burdened by one particular issue. You know, there is a history of 20, 25 years of cancer vaccine T-cell trials. They all have failed. There has not been a single success story. Why? Because we did not yet know how the immune system works to create balance between building an immune response and then stopping that immune response. So whenever a cancer vaccine was used to build a T-cell response, the immune system has a natural mechanism to stop that response when it gets too big. And for cancer, you need a really big response to eat away those tumors. So vaccines haven't been successful for T cells because the immune system naturally stopped those responses. Even if the vaccine was actually inducing it, it didn't continue to build. So now that we know what checkpoint modulation is, which basically means you can take off the brake from the immune system and let the immune response build further, we're now testing, and that's what all these trials are about, we're testing if checkpoint modulators, which are antibodies, can be combined with those T-cell vaccines. That's why there's so much activity. Now, what's a very small fraction of this is actually B-cell vaccines. And the reason for that is not that there's no interest or confidence in B-cells. We know they work as an immune mechanism. It's because the field is so young. We only had 6 years, from 2011 until now, to get away from the T-cell story, which is predominant in the immuno-oncology space. With the last 30, 40 years since we're trying to use the immune system, we always focused on T-cells. So now that there's the first success story was with T-cells. So everybody wants a piece of that success story, so everybody's using T-cells. And we're now just getting to the point that we need new things. We've shown how broad the T-cells can be used. We need something else. And the immune system offers the opportunity to use B-cells. So people are starting to come around and look at B-cells as new opportunities. And I think Imugene is at the forefront of that next thing that's coming. So there's very little competition right now, but I expect there will be a lot more as we go forward.
Thank you very much indeed to the audience and obviously to our panel. I'd now like to hand over to Leslie for a few closing remarks.
So if you have any dying questions, we will be around for the next hour or so you can approach these oncology luminaries for further questioning. So I just would like to thank our three presenters here who travel far, and the experience that they bring to me and to Imugene has been immense, and I hope that you were bestowed some of that honor as well. I just want to say that all of us at Imugene look forward to updating you all as we progress into our current clinical trial in gastric cancer and the many strategic initiatives we are working on to expedite this valuable technology into the hands of people who need it, which is our patients. So thank you so much for coming.